Right, thank you. Just like to start by thanking Andrew and the SciDab crew for the invitation to speak. I'm actually here on behalf of Roby, who many of you have possibly met before. He does send his regards. He was unable to make it this year. So yes, I'm going to be talking about basically looking at maybe the mistakes of the past and then doing everything we can to ensure that we don't repeat them in the future, because there's a brave new world of data coming towards us. So first of all, CST, it's a privately owned company in Danvers, just north of Boston. We've been around since 1999, have about 600 people. And our mission since the beginning has been to provide the world's highest quality and innovative research tools to accelerate biological understanding. And being that is our mission, it is extremely gratifying to be recognized by organizations like SciDab for the quality of the tools that we have provided to the research community over many years. These two awards have really meant a lot to us, but also innovation is very important to us. We actually consider ourselves an R&D organization, a true research organization. And so to be recognized for innovative developments in this space, which is becoming increasingly commoditized, is very rewarding to us. So thank you for that. And this innovation award was in regards to a portfolio of reagents designed to detect engineered CAR T cells, which have been very enthusiastically adopted by the CAR T community. And so core to our success at CST is our innovation, but also our commitment to reproducible data in science. And this has been something that we've taken to heart since the very beginning. And just like with the SatNav satellite, which is trying to identify the location of its target, which it knows is a set distance away, if you only do one measurement, you can only really estimate the target location with low precision. However, if you do another measurement with another SatNav satellite, you can increase your precision significantly. And if you introduce a third measurement, you've got very high precision in identifying your target. And so if we think about target precision in our space as being the target specificity of an antibody, then these satellite measurements really are validation experiments. And so you need more than one validation experiment in order to really demonstrate target specificity. And so we strive to always perform multiple validation experiments to validate our antibodies for target specificity. We don't rely upon one measurement, but multiple measurements. And a framework for doing that are the hallmarks of validation that CST did not develop, really. I mean, we built these off of the 5 pillars of antibody validation that were proposed by the International Working Group. So they are very similar, but not identical to those pillars. And again, we use this as the framework for our validation. But there's more than that, because antibodies, of course, are used in a number of different ways or applications. And so our goal is always target specificity, specifically within a given application, a fit-for-purpose approach. And so these hallmarks of validation are applied independently within applications. It's always great to have correspondence in your validation hallmarks across applications, but that's not necessarily always going to happen. So you must interpret in the context of the use. And I'm just going to summarize some data, first of all, describing our approach for immunohistochemistry validation that we described in a special issue of the Journal of Histotechnology in 2020. This was a special issue on validation and reproducibility in histotechnology. And this is looking at validation of 2 antibodies against 2 very similar targets, YAP and TAZ. And this paper really seemed to resonate with the audience. It was viewed over 3,000 times after publication and is actually the 7th most cited paper in the journal's history. And so this reproducibility message really seemed to resonate with the audience. So YAP and TAZ are transcriptional coactivators in what's known as the Hippo pathway. They have very similar sequences. They are homologous proteins. They operate in similar biological pathways. They have similar mechanism of regulation, and they have similar expression in cancer cell lines. And so they're frequently referred to collectively as YAP-TAZ. You'll see that in the literature over and over again. They may be talking about one or the other, but they refer to it as YAP-TAZ, leading to the assumption that these are actually almost identical or redundant proteins. But we didn't really know that for sure, and we couldn't determine that without actually being able to interrogate their expression specifically. And to do that, you need specific antibodies. And that's specifically in the context of tissue biology. As I mentioned, in cell lines, they're expressed almost identically in different cell lines. So using our hallmarks of validation, we set about validating antibodies for IHC for both of these targets. We, of course, this is not in sequential order and it's not in priority order. It's just representation of the work that we did. So we looked at binary expression models, and we actually did 2 of these. We first of all looked at cancer cell lines where we know there was positive expression for both YAP and TAZ, and that would be SKMAL28 as one example. And then B cell lines do not have the Hippo pathway active, and so RL7 is an example where there's no YAP or TAZ expression. And these antibodies, these 2 clones, using the candidate clones that we were working on, showed expression in the positive cell line, but none in the negative. Now, of course, these are not isogenic, so we moved on to a knockout cell line. Now, we didn't have single knockouts of each of these 2, but we did have a double knockout cell line, and you can see that in the 293 T cells, there's positive expression for both YAP and TAZ detected by the candidate antibodies, but nothing in the double knockouts. Now, of course, this does not convincingly demonstrate that the YAP antibody does not detect TAZ and vice versa. So we moved on to a heterologous expression model, and we overexpressed YAP and TAZ independently in cell lines that don't express YAP and TAZ. And as you can see in this panel, the YAP transfected cells are detected, their signal is detected by the YAP antibody, but not the TAZ antibody, and vice versa in this. So we demonstrated pretty convincingly that the antibodies are specific to their target. Now, this is only in cell lines up until now, so then we move on to tissue, because you cannot approve for immunohistochemistry without actually testing in tissue. So we looked at a ranged expression model. This is actually combined with an orthogonal model because we're using messenger RNA data from the Human Protein Atlas as our reference, and we selected tissues that are high in expression for YAP, medium level expression YAP, and low expression. And then we did the same for TAZ, although I'm not showing those data in the interest of time. And when we show that you can indeed detect high levels of signal in smooth muscle, which is the high tissue, moderate levels in kidney, which is the medium tissue, and low levels in cerebellum, which was the low level of expression. So we saw perfect correspondence, and then we saw the same thing with TAZ, as I mentioned. And then lastly, moved on to Matthias's favorite strategy, the multiple antibody strategy. And luckily, we had multiple clones in development, all of which we'd already validated for Western. We knew that they were appropriately specific in that assay. And so we moved on, and we have a polyclonal YAP antibody that's been IHC approved for many years, so we use that as our second antibody, and we observed identical staining across tissues with the new monoclonal antibody compared to the poly. And similarly with TAZ, we had secondary clones that were also positive and specific for TAZ and had evidence of functionality in IHC. But again, these are different epitopes, and so in both cases, we were able to show completely corresponding staining across that. So collectively, all of these data together, these multiple hallmarks, led us to be confident in the target-specific validation of each of these antibodies in tissue. And so then we moved on to characterize and looked at the expression of YAP and TAZ in both normal tissues and in cancer tissues. And we were able to confirm that they are indeed not redundant in terms of their patterns of expression. There's very different variations, and it differs from tissue to tissue. And that's true in both normal and cancer tissues. So leading us to conclude that YAP and TAZ are not equal to YAP-TAZ. They are indeed independent proteins that should be considered in isolation. And until the availability of antibodies, that was not a question that was not able to be answered. I mentioned application-independent validation. This is just an example from another project. This is Claudin 6. This just illustrates the point that validation in one application does not indicate its approval or validation in another application. These are IFIC data from a clone E2S5N in a positive and a negative cell line. You can see that there's nice positive membrane expression in the positive cell line and nothing detected in DU145, which is negative for Claudin 6. When we look at exactly the same clone in FFP fixed cell pellets, exactly the same cell line, you actually see signal in the DU145, ruling out that clone for further testing in IHC. We've stopped the validation right there and said this is not going to work in tissue because it does not even work in cell lines. Luckily, we did identify another clone that was clean in the DU145, and then we went on to further validate that in tissue using our hallmarks, and that is why we have two clones in our catalog, one of which is validated for IHC, and one of which is validated for IF. And so that means something. It's very important on the CST product pages. That recommendation comes with validation data behind it. That moves us on now to the next frontier. So this is, all of this is relatively, the data I've talked about is really not new. This is talking about the standards that we've been talking about for years now. But now we're moving into this new frontier of spatial biology, which I'm going to talk about in the context of multiplexing. So just to get on the same page, defining spatial biology is understanding target biology within the spatial context of other targets. So typically this is in a tissue sample. It's fundamental for true biological understanding, and it typically involves observing multiple targets within the same sample. So then this introduces a lot of challenges. These include technical challenges. There's a lot of different protocols that could be used. There's instrumentation, a lot of different platforms that may be used for this. There are computational concerns now with interpreting the data that you are getting. And of course, there is the validation of the reagents that are generating the data, which again must be done in a context-specific way. This graph here just shows you the explosion in interest in spatial biology. This is simply a PubMed search for the term spatial biology and research, or spatial biology, showing you the interest in this space. And it's these type of data that I'm talking about today. So it's looking at multiple markers within FFPE tissue samples. So the message that I'm trying to get across today really is that validation needs to consider both sample and assay complexity in the context of reproducibility. So if you're looking for an antibody that's simply a target binder in a simple sample, perhaps it's a walk in the park in many respects, an overexpression model, for example. If you're looking for a more complex assay, the challenge gets a little bit more difficult. If you're looking in an IHC context that I just described, of course, this is among your most complex sample types, so your challenges are getting significant. Now we're talking about multiplex IHC. So the challenges really are astounding, and I think if you moved into the clinical space, that's probably the Mount Everest picture that I did not include here. So I think that this increase in sample and assay complexity is increasing the number of variables that are at play and therefore increasing the challenges in reproducibility. And we need to be thinking of this as we move into this new era. So some of these variables that I'm talking about include the initial validation of the core reagents that are being used. So you need to have, if you're looking in tissue, you need to have IHC validated reagents to start with before you even begin modifying them for a spatial context. There's typically going to be conjugation involved, so there's different chemistries involved in conjugation. That needs to be taken into consideration. If there's an amplification method used for the signal, that needs to be taken into consideration. In addition, the sequence or the order of which antibodies are added, that is a variable that can be depending on the platform that you're using. Methods of stripping and removal of either the antibodies or the fluors is another variable. The abundance and the target variability amongst tissue samples needs to be considered because the order of the antibodies may actually influence the detection of the signal. You now have the opportunity for interference between epitopes. Does one antibody block another antibody? That needs to be considered. And of course, the protocol that you're using in general, that includes things like, are you using a manual protocol or an autostainer? Other protocol variations. These are all things now that can influence the outcome of your experiment. And so what I'm going to be talking today just is our foray into this space now. Just this week, we launched a multiplex platform for IHC called SignalStar. It offers the opportunity for doing a 3 to 8-plex fluorescent IHC assay, so that's a mid-plex range. It has signal amplification included, so that helps for low abundance targets. It does have cycling, but there is no epitope loss because we don't actually remove the antibodies. And it's validated in both automated and manual protocols. And importantly, of course, we've done extensive validation of this platform on FFPE tissue arrays, both normal and disease. And so the platform rests on oligoconjugated antibodies, and there are essentially two, for an 8-plex, there are 2 cycles for this. The first step is to have you fix tissue and add all the antibodies at once, so there's no sequential addition or stripping of antibodies. Then you amplify the oligos with matched oligos for the first 4 targets. You then image, and then there's a removal of those oligos, which have the fluors on them, but the antibodies stay there. And then you add your second set of fluorophores amplify, and then image your second round. So if this is done in the same 4 colors in the second round, but then you pseudo color, so you get 8 different colors. And so being that we're using oligoconjugate antibodies, there are some new validation requirements, and I'll just describe some of these to begin with. So again, using the framework of our hallmarks of validation, we first of all always confirm that the antibodies are validated for IHC before we begin conjugation. And then we look at post-conjugation and make sure that they're still functioning in an IHC context. We specifically use site-directed conjugation because that's a way that we can control the conjugation chemistry. Instead of having random lysine-based conjugation on your antibodies, we know exactly where and how many oligos are added to the antibodies. So we control the degree of labeling, and then that allows us from lot to lot to provide a reproducible reagent. And so just some examples of the pre and post conjugation validation. We of course use the reference IHC as a gold standard, and just here's some data here showing 4 different markers where we see identical signal between both the oligoconjugated versions and then the original antibodies with FFP, with DAB staining. And we try to quantify this, right? So we've gone through and we look at every antibody and we put it in a different location in a 4-plex panel and then count the number of positive cells, and we compare that to the DAB stain, the traditional IHC, and we see correspondence, and regardless of which round you put it in or which floor you used, we get similar results in each time. And so we end up ultimately able to generate these beautiful multiplex images like this. What's amazing about this, we believe, is that the entire protocol can be done in 2 days. So that's a significant time savings with compared to current opportunities, other options. So our conjugation chemistry is also unique. So it's site-directed, as I mentioned, and we optimize for the degree of labeling. So we use size exclusion chromatography to determine how many oligos are added to each antibody, and we optimize based on the degree of labeling. So then once we know if there's 3 oligos or 4 oligos, which gives the best signal, we then always choose that for subsequent lots of the antibodies. So each of the floors antibodies that is conjugated is optimized for degree of labeling during validation. And it does make a difference. And so we compared our conjugation chemistry with a more typical conjugation chemistry, and we are able, because we control the degree of labeling, which allows for you to control the amount of amplification, you can get actually significantly more signal to noise, even at the lowest antibody concentration, the same antibody here, but with 2 different conjugation chemistries, we get higher signal to noise at the lowest, a lower concentration of antibody than the comparable chemistry. And lastly, I mentioned imaging round. Its flexibility is something that a lot of people want. They don't necessarily want to have to do things in a prescribed order. And so we've tested all of the antibodies that are in our panels in different order. You can add them in the first round, the second round. Doesn't matter which floor you want. You can essentially build the panel that you want and then and use it as you desire. So there's no antibody removal between the rounds because they're all added at once, and it's only the floors, the oligo floors, that we're actually removing and then binding. And so lastly, there are both manual and auto stainer protocols, and we've done the validation to demonstrate that you get the same results regardless of which protocol that you use. So we support protocols on the BondRx. So I'll just wrap up. In the multiplex era, we have a lot of challenges facing us. The guiding principles at CST have always been that you can't validate with a single assay. You need to do multiple validation experiments to demonstrate specificity. It must be done in an application or fit-for-purpose way. And validation requirements do scale with assay complexity. And so we think that multiplex technologies have enormous potential to reveal new biology and advance the field, but rigorous validation to consider all of these new variables is absolutely essential if we are not to repeat the mistakes of the past. So thank you very much. I'm happy to take any questions.